Hi, hello everybody. Welcome back again to this channel, One Question a Day, in answering all histology and tooth morphology questions. The question that we are going to discuss is the histology of mixed salivary glands. First, begin with the outline of the salivary gland. Mixed salivary gland, it consists both serous acini, mucous acini. Not only that, we have other structures. They have an outer connective tissue capsules, which you give an idea. Connective tissue capsules, you give an infographic of mucous cells, the septa, the lobes, the serous cells, the serous demuluns. An outer and a histopath diagram will be greatly appreciated. Normal histopathology diagrams. They have an outer connective tissue capsules. The invagination of these capsules or a septa forms the numerous lobes. The lobes, each lobes consists of numerous serous cells, mucous cells, demulunes, and collecting ducts. The serous cells are pyramidal shaped, as their nucleus spherical. A lot of organelles, specifically zymogen granules, the Golgi apparatus, rough endoplasmic reticles, and numerous vesicles. Whereas mucous cells, you find broad columnar shaped, flattened nucleus, very close to the basement membrane. Nucleus is not along the basement membrane, but close to the basement membrane. And you have very less zymogen granules. Literally, no zymogen granules are visible, but there are granules. The examples of mixed salivary glands will be the submandibular glands, sublingual glands, labial glands, and part of lingual glands. The specific characteristic what we see is the demulunes, which is present in the exclusive to the mixed salivary glands. They are essentially mucous acini surrounded by a C-shaped serous acini. People believe that this is an adaptation. The nature of salivary secretion, they can be serous or mucous, predominantly a mixed glands. And how it works in a mixed salivary gland is parotid is increased watery, has increased amylase and increased proteins. Submandibular has increased glycosylated substance. Sublingual glands has increased mucin substance. The nature of stimuli, rate of flow as compared to the stimulus of rest determines the seromucous nature, intensity of the stimuli. Day has a more serous, night has more mucus. So all this combined to form the sympathetic and parasympathetic stimulation modulation, and the stimulation of the serous mucous salivary acini determines the amount of final salivary product. Clinical significance, inflammation or injury to salivary gland may to lead to cysts such as mucoceles or salivary stones that can block the saliva. Infections such as mumps can derange the salivary acini. So, mixed salivary glands are known for their ability to form tumors. So, don't make this mistake of putting the zymogen granules. Draw histopathology diagram, neat one. Stay connected with this channel for another question answer session. Till then, learn incrementally. Happy learning.